Thank you for joining this talk. And today I will present some of the work that we do in my lab, in my team. And the title of my talk is Investigating Drug Effects Using Phenotypic Cell Profiling, AI and Lab Automation. So my name is Ola Spjuth. I'm professor at the Department of Pharmaceutical Biosciences at Uppsala University. I also contribute to one of our spin-off companies, Scale Out Systems, which I will mention in this presentation as well. So robots and automation are making their way into our labs, and we believe that they will be increasingly important in the future. This is a video of a moving lab robot. It's at Fraunhofer, and it carries out some automated tasks. At the same time, I also believe that AI will have high impact in the design of experiments and also in hypothesis testing and generation. Today we use them mainly to make predictions, but this is something that I believe my team believes is coming. And we also, where we today study, looking at fixed time points, maybe you look at, a time point 0 and time point 24 hours later. There's so much in the temporal profiling where we look at things over time and adding the time dimension that there's so much data that we're not using today. I think this is going to be increased a lot in the future. And as we increase and move more into the temporal domain where we continuously collect data on biological processes, we will undoubtedly have an increase in data velocity. And where we now have sometimes a disconnected data collection and data analysis step, I think that these will have to be merged in the future. So these are some of the basic foundations that we work upon in my lab. And it's been a lot of publications and a lot of adverts about how the increase in data is sort of changing the way we do biological science. This is just one of the one presentation that shows this. So who are we? So I'm leading an academic research group at Uppsala University. We have a background in computational pharmacology. We worked a lot in data science, AI, machine learning, a lot together with pharmaceutical industry, and worked on predictive modeling and machine learning for over 15 years. We're also good at e-infrastructure and big data, so the data engineering part. It's not only about the analysis, it's about how you can have an infrastructure and work with large-scale data in an efficient way. And we've been part of several Nordic projects and European projects and accumulate a lot of expertise in that. And now we're setting up an automated high-content imaging lab for cell profiling. And I will talk a little bit in this presentation about how we try to put these pieces together. And the research objectives in my team is that we would like to accelerate drug discovery with AI automation and the design of experiments. And we're mainly looking at predicting safety concerns for early in the drug discovery process, trying to explain drug mechanisms, but also screen for new drugs. So traditional hypothesis testing usually comes from where there's a human. The human forms some hypothesis, devises some experiments to carry out, and then it goes back to this human, the scientist in the center, that does the analysis and the interpretation. And this might lead to more experiments, or it might lead to the scientist revises the hypothesis and do more experiments and so on. And ultimately, hopefully, this process leads to some sort of insight. So how can we look about this process? Well, it's clearly iterative. It's extremely flexible with humans carrying out experiments and doing analysis. We can more or less do everything that we can. It's mostly manual though, and it becomes rather slow. Whereas in predictive modeling, this data collection step has usually been that you have some samples, maybe you have large-scale samples, and you analyze that, and you produce a database, and then the modeling, the prediction is sort of separate, and there's usually still a human doing query to an existing database and get some where maybe potentially train some model and do some prediction. So what do we observe about this methodology? So, well, it's mainly retrospective. You generate data that you hope will be predictive in the future, and in order to do so, usually you would have to do large-scale data, because a lot of the things that you do will not be particularly informative. And doing large-scale things, maybe with automation and other things, is quite expensive. And also, in the end, it's relatively limited for hypothesis testing. It could definitely be so, but if it doesn't work, well, there's no real way of sort of, if the model doesn't produce, so what do? You can't really get more data in this model. So, data-driven science, there's a lot of hype on data-driven science. So what do I think this is another way of phrasing this, the previous slide, that a scientist has an hypothesis and sort of uses his or her intuition and experience to devise an experiment that leads to data that could sort of be modeled into insights. But we'd like to go a little bit more that, the data in themselves drives the hypothesis, that the data in itself drives the design of the experiments. What should we do? And that we also can make better use of data, and also that data and compressed forms of data in terms of models also can sort of lead to different types of hypotheses. So this is how I would like to, this is what I'm working to trying to achieve in my lab on a more methodological level. So our vision is really this autonomous experimentation or closed loop experimentation, that we have existing data and we sort of combine it with external data someone else has produced. It could be public or collaborators. We have a continuous analytics AI system that sort of compresses this data and makes it possible to ask some questions around it, and it also can devise the next round of experiments. And if we have adequate automation that can carry out these experiments, well, we can close this loop. But of course, this is it's a complex thing, and right now we're working a lot on trying to get the automation in order to do something in this, and also at the same time build up an informatics platform to handle all the data analysis that has to be done. But if we look at the considerations for sort of the next experiment, I think we should honor quality over quantity. Better data is often more useful than simply more data. There's a lot of hype. We need large scale data. Yeah, but you need large scale, high quality data. Just large data is not going to be enough if it's crap. I can very easily generate a lot of data that's just random. And we also have to acknowledge that data collection may be expensive. There's a cost of the time and the materials for experiments. Some data might be more expensive than others to do that. And raw data like an image can be very easy to produce, whereas an annotated image where an expert has looked at it, identified interesting regions, could be much more time-consuming and expensive to do. So what we want to do is, of course, to collect the best data at a minimal cost. And in our cases, in most of the objective is usually to improve the accuracy or confidence of predictions in an AI models. So the fundamental question then becomes, can machines or AI learn with fewer training instances if they ask the right questions, instead of a human deciding what data should be captured? And active learning is doing exactly that. If you compare it to passive learning, where you get some data from nature and you train a model, in active learning, the learner puts a query to, in this case, nature, and gets a response back. And it's an iterative, you build up a more predictive model. And the hypothesis is that by querying the nature in a more intelligent ways, a more rational way, you can sort of reduce the number of training examples to get the same accuracy of the model. But of course, it's a very hard problem that we're trying to do. We're trying to explore it, unchart the unknown, explore unknown things. So there's a sort of a trade-off between exploration, where you go where you have not seen anything before, that could lead to better predictions in the future, or the exploitation, if you go to a more interesting area where this area is interesting and you want to make the best predictions given these things. So there's a trade-off, and that's always like that. So switching over to automation in life science. I mean, that's sort of, there's a varying degrees of automation when you talk to different type of people. For example, you could have an automated instrument that works with a microplate, and we will consider this. Yeah, this is a level of automation because I put it in the instrument, and then the instrument sort of, works with all these, 384 or 96 wells, for example. Maybe you have a stack of microplates you can put in an instrument, and it sort of processes them in an automated fashion. And by saying a robot in life science, most people probably refer to a liquid handling robot. So that, like when you see here on the picture, when there is a robot carries out a series of steps of pipetting and potentially can move things around within a limited scope. But an automated lab can also be a set of instruments where they work with, for example, microplates, or it could be a plate handling robot serving multiple instruments, and you have these work cells that you can buy if you are rich, or if you work at a company that has a lot of money, and where you can perform integrated analysis. So automation spans a large gap from having a microplate reader towards these full-blown installations. So a great inspiration for me and myself has been the robot scientist concept, which was developed by Ross King et al. And they published their first paper already in 2004, so it's quite some years ago. It was updated. I think it's the automation of science in 2009 was sort of is probably the core paper in this sense. And this was sort of he built a robot that could do integrated analysis, and it was sort of was able to hypothesize, do device experiments, run the experiments, and interpret the results, and repeat the cycle and so on. And they demonstrated that they could acquire new knowledge using this. I think it was in yeast or in the beginning. So this has been a great inspiration for me. And another inspiration has been the field of what we now call phenomics and high-throughput biology in this sense. So Ann Carpenter, I think, pioneered the image-based profiling in this sense, but the field is larger than that. And I mean, it's sort of, we look upon this field that we work with cells and profiling cells. So we have various type of cell lines, and we could sort of, engineer them in various ways, or get them from patients, or having stable cell lines. And then we can perturb them in some way. We can, for example, expose them to molecules or some sort of interference. And then we measure, so how do these cells change their phenotype? And we can do this with molecular profiling technologies like transcriptomics, proteomics, metabolomics, and these things. And we can get a bit of profile, what type of genes change due to this perturbation. But what we are doing is more morphology profiling. So we're looking at how do the cells change their morphology when they are sort of exposed to some sort of perturbation, such as treatment to a drug. And we then capture cells out of this. We can cut out the cells using image analysis and quantify this in various ways. And we can get some sort of feature out of this and that we can use. And in a similar way to, for example, let's say a gene expression profile, we get an image-based cell morphology profile here with a feature of numbers that we can use for computational analysis. So cell painting is the core protocol that we do. So in principle, it's a relatively simple idea that has a lot of power. So we have cells, we expose them to some sort of compound usually. We do that in multiplates, in stacks of multi-well plates. And we stain them, for example, with 6 different dyes, and we image them in 5 channels. So every well, we take 6 different sites. So that would be 6 times 5. So 30 images per well, per time point. And we do this in 384 well plates, and we do it in stacks of them. So you realize it's a lot of images. And if you add the temporal dimension, it's a huge amount of images. And based on these, we can then if you have a few hundred cells that you cut out from each well, well, you get these features. You can quantify them into features and do unsupervised and supervised learning to this. But what's shown to be very powerful is to use these images in convolutional neural networks. So of course we use that. So then we don't have to sort of segment the cells and cut out the individual cells. Then we look at them as more we're sending in the raw image in a neural network, and then let the algorithm then define the features, and we can then make predictions with high accuracy. This is one example of what we have done on data where we expose cells to different types of drug compounds, and when the compounds come from different classes, and then we see, can we distinguish between these classes? Can we predict these classes based on the images? And yes, we can. With high accuracy, we can say that these compounds sort of fall into the different types of classes in this sense. So, and this technology works, and there are pharmaceutical companies, if you've heard of the Recursion Pharma, they've been quite hyped recently. They work a lot on, this is their core methodology, where they develop a cell model that has some characteristics of a disease, and then they screen and see, can we revert this phenotype back to the normal phenotype using drug screening, and that's a phenotypic screening. Here's another experiment that we have done, where we look at combinations of toxicants and how they affect the cells. We can see, and this is a small experiment for us. It was just 3 cell lines and 3 compounds, but we look at them in all combinations and all concentrations, do that in triplicates and 2 time points. So this amounts, this small experiment that you do in a few days is still 16 plates, leading to over 40,000 images or 400 gigabytes of data. It's a lot of images that needs to be processing this, and this is a really small experiment. And in the end, it's sort of, you can pose some questions around this. We're still looking into this and running some follow-ups on this. But the image to the lower right here shows when we cluster them in PCA space, we can see that we can clearly separate the compound H from the compound D. The compound D looks very much like the control in this sense. And you see the combination when the cells have been exposed to both D and H. You can see that they cluster together in somewhere in between D and H here. Well, there will be more coming out of this. So another project that we're doing together with AstraZeneca in the project is to look at lipid nanoparticle-based drug delivery, and where in this case they are creating lipid nanoparticles to deliver some cargo into the cells, and they want to sort of create a more targeted delivery, and so that they should only fuse to certain types of cells. In this case, they are delivering the cargo in terms of mRNA for GFP, so that if the cells sort of, if the LNP is sort of taken up by the cell will start expressing GFP. So you see on the left side, you see the in orange color are the lipid nanoparticles, and then in the middle part, after some time point, you see some cells have started to take up these particles, and some cells are now on the right side beginning to express GFP in this sense. So apparently some cells take up this lipid nanoparticle and others don't. And then you can vary a lot of things on top of this lipid nanoparticle to sort of custom tailor them to fuse with certain types of cells. So we wanted to work with the deep learning models that also were dynamic, so that we take into the time dimension into account, and see if we could sort of predict this happening without having to run the entire experiment. So this is an example of a slide of 6 different wells in one microwell plate. When you see this over time, and you can see that the nanoparticles start to go around, and in some cells you see the lipid nanoparticle have been taken up. So in the lower right here, you see almost complete uptake of the LNP particle. A lot of cells that are showing expressing GFP, whereas in the upper left, you see that no GFP expression, and on the upper right, you see some GFP expression, but also a lot of the LNPs have clustered together in this sense. So that was just an experiment showing how it's done. So while doing this, you end up with, okay, so cells are starting to sort of, express more and more GFP over time. But then what happened to this cell? What happened to it? Did it move away? And you look at it, and you can cut out these individual cells, and you can see what happens to this cell. Well, apparently this cell decided, I don't want to live anymore. So you see, it commits suicide and doing by apoptosis in this sense. That's just a sidetrack. We have many cells. You look at many cells. So some of the results that we have recently published in a bioarchive paper that's been submitted is then to compare then the question we wanted to ask ourselves here, when we look at this with traditional prediction model, we look at the one time point against the other. But what if we take the temporal dimension into account, so that we add that with LSTM, a long short-term memory model, and we see that including the temporal dimension, you see that with the pink bars, they are consistently higher F1 score for predictive power than if you look at the traditional CNN method. And on the right-hand side, we see the previous. On the left-hand side, it's a classification. We said something like high or no, low. And on the right-hand side, we look at it as a more regression of that. We try to predict, for each individual cell, will it predict GFP, or will it start to express GFP or not? And even here, we see advantages of including the temporal dimension in this. Here's another project that we are currently doing then. We're looking at coronavirus infected cells. This is a SciLab Lab funded project in the drug discovery and repurposing of drugs research area. And it's a collaboration with several partners, some at KI, some at Karolinska Institutet, and some at Uppsala University and the Chemical Biology Consortium. So what we wanted to do then was to look at coronavirus infected cells with multiple profiling technologies. So we'd like to sort of have standard cells, infect them. We started with the 229E strain. Now we're doing the SARS-CoV-2 in a BSL-3 lab. Then we do different types of drug treatments. And then we do profiling. And in my lab, we do cell morphology with profiling. And in the Sven Erlander lab, we do transcriptomics profiling with a drug sec. And the aim is, of course, to do this integrative analysis. We haven't really gotten to that yet, but we've set up a lot of the methods to do this. And I have to click here after I do that. So here are some, for example, here are some of the reasons that's where we look at, when you look at coronavirus or other types of viruses, you have to sort of see, do they enter the cells? And I said that previously that we color cells we can read them in 5 channels. So you have to sort of sacrifice something here. So usually we color the nuclei, nucleoli, ER, actin Golgi, and mitotracker. But here we decided to sacrifice the mitochondria, the mitotracker, and instead color the coronavirus. And here we have a result for 3 different cell lines where this works. And you can see that some cells have been infected and others are not. So we can now try to sort of, now we're moving to the next step when we're doing the drug treatments. But what we can do is that we can, within the same within the same sample, we can look at how different cells are affected differently by a drug treatment. So because we can single out what cells are infected in this, and I think it makes it a very powerful approach that 2 cells in the same experiment can be looking at. We can study their drug interactions using cell morphology on single cell basis in this sense. So I'm looking forward to do this, and of course we want to integrate this with transcriptomics a little bit later, but that will obviously be for now it will be on the population level. But going back to our lab, our aim is to do an intelligent system for chemical profiling, usually with drugs, that we can, the system should design the experiments. We have the automated lab to carry out the experiments. We want an automated pipeline to do it. We want to publish data and models and iterate these things. And we have in our wet lab, we can also sort of verify this with, you know, external protocols in this sense. So we can test and generate some hypotheses. So to me, this that what we're building up is a bit of a discovery engine in this sense, and we're looking into different types of topics, like looking at efficacy mechanism, toxicity combinations, but also screening. So that, but it's based on our the methodology of cell profiling. Now we do fixed cells. Maybe we'll do live cells, as I showed later. It depends. But the interaction between robotics, AI, and this type of methodology. So what do we do then? Well, we need a fully automated cell painting so that we can do this. So the robot can do everything. And then you need it's taken a lot of time to build this up with facilities, environmental control, ventilation, and procuring instruments, automation systems and protocols, but also not to neglect the infrastructure in computational resources and the software and pipelines. So we started with a fixed setup that was available at the department. We helped set this up. So it was a procured system from a vendor, Molecular Devices. It was a plate robot that could move plates around, the plate incubator, a liquid handling system, and some automation software. And it was pretty slick to get up and running. And they always wanted to, yeah, let's fix the protocol to do that. But we wanted it to be a little bit more customizable by us, and that was a bit of a problem. It was not really easy to expand or configure for us. Our priorities are really the flexibility. We want to sort of, you know, expand this over time. We would prefer to work with open source so that we can, you know, code the different parts of it. It should preferably be low cost, and we should be able to service it ourselves, because it's very expensive if you have a company coming to service these robots, and configurable. So instead we started to set up an open source lab automation project. We procured some instruments and built this up. So we've now set up this system, and we're working on the scheduling system, and we put all our code on GitHub, so anybody can sort of use our software. And we're putting this together. We really would like to have collaborators on this. If you're interested in lab automation, or then just please contact us. So this is an image of the lab that we have built up. What we have done is that we've sort of we've built all the components. We have wrapped with our own software. So it interact with the washer has its specific software, and we interact with that software. But the whole scheduling system is completely open source. And then we sort of course, have automated moving microplates around and do experiments in this sense. So I'd like to say something about the dealing with large-scale data in this sense. Since we're preparing ourselves for huge amounts of data, we've sort of built up an infrastructure to do that. And so we have our own on-prem solution. We have some hardware. We have some CPUs, some GPUs, and some storage, a few hundred terabytes. And we run everything in virtualized environment with Kubernetes, software containers, where my group members, they like to work in TensorFlow a lot, and some use Spark and so on, others. And now what we are really working on and trying to put into production right now is to, when the robotized lab and the microscope spits out images, that we can automatically process them using QC workflows, automated calculation of these profiles. And we also want some interestingness models so that we don't store uninteresting data. We just can't keep that. So that's something that we're trying to put some work in developing as well. If nothing has changed between 2 time points, should you really store that image? We put all of these as open source on GitHub. We developed some image viewers and a database to store these results. And we also have for our data scientists, then they can then use these databases and the data produced in Jupyter notebooks, for example, where they can do some coding. And when they work in these notebooks, they also have access to GPUs so that they can do AI modeling and produce some models. And if you look at the lower left, we can do this first. We publish these models as well so that others can use it outside of it. That's one key important for us. But on the lower left side, you see, we would really like the models to sort of then guide the robotized lab in the next round. What experiment should be done next? We're not there yet. We're getting there. I'm hoping we should be able to put this in production next year. So data is not static. We will continuously have more data coming in. So we continuously need to retrain our models. And this is actually a practical problem. I think most common right now is you produce a data set, someone else takes it, someone else does the data analysis of it, and then the data analysis is done. Maybe you publish it in a paper and it sits still. And that's not really going to work in the future if we build up a system like this. So we worked a lot on how we can automate the AI modeling lifecycle, and this is where we're interacting with the company Scale-Out, which I mentioned before. I've gotten involved with, and also the Scale-Out Lab Data Center, where we want to really build up an environment where if you've trained a model, that you can sort of package and deploy it so that you automate all the steps, all the pre-processing steps, all the training steps, the QC steps, that you have proper versioning of your models. And we're working quite a lot on this to get that running in a nice way. And sort of wrapping up here, there's a high synergy with what my team does and what we contribute to this project called HASTE, which hierarchical analysis of spatial and temporal data. That is an SSF-funded project where Carolina Välby, professor at IT department, is the PI, and me and Andreas Hellander, also at IT department, is PI. And then here we're really working on many levels on how we can sort of, when an instrument produces a lot of data, can we online classify it and use policies so that we can only store the most interesting data. And we're working with AstraZeneca and the company Veranova, and it's usually it's microscopy and large-scale imaging that comes out of this. And we really cannot store everything. We just cannot do it, and it's not useful to do it. So how should we prioritize? That's the kind of problem we try to solve in this project. So we developed some software, and this is free for anyone to use. I hope it's useful to others, but we built it for ourselves. Our infrastructure, our virtual infrastructure with Kubernetes, is available as a code, so you can download and run it. The database with projects, images, and results is already run by, and the image database and the image viewer is already run by another group at KI, because they wanted to have something to look at the images from high content imaging. The batch and continuous cell profiler pipelines that we do, I think they would be very useful for others. We're putting them online. Some of our, when we do deep learning, we do notebooks, and if we do a manuscript, we usually publish the notebook so that other people can sort of reproduce that. Our open source lab automation system is in progress, in development. It's also on GitHub, and I really like the project that we're are going to submit relatively soon, where we design the cell-based experiments more efficiently. Where you sort of, what experiment should you do? I mean, if you decide, yeah, I want to try these 50 compounds in the next round, how do I design them? How do I put them in microplates in the most efficient way? And we're using constraint programming to do that, and that's a really cool project. Yeah, compound annotation, that's not really started yet. Of course, we would like to integrate it with our other AI services. We have done previously a lot based on chemical structures, where we predict targets based on chemical structures, and also site of metabolism, these type of QSAR-based studies when you come from structure activity. We want to integrate that with our cell profiling as well. And I was really happy the last week, because the Swedish Research Council, they funded the project to really build this autonomous genomics lab, where we have different types of monitoring using deep learning, active learning, other types of predictive modeling to sort of design the next experiment and sort of close the loop in this sense. So we're going to hire people to this project, and that makes me very happy. What we're working on is our cancer. It will be cancer and sarcoma cells. So we'll apply this system then to try to predict new drug combinations, first on cell lines in the step 1 to the left, seeing the difference between sarcoma, non-malignant cells, and co-culture. And we'll in step 2, we'll try to sort of come up with new drug combinations that are efficient for these cancer cell lines. And in phase number 3, we get the cells, primary cells from patients that have these sarcomas, and we then use this autonomous system to calibrate our findings. If the 1 and 2 are done on cancer cell lines, the third one is to calibrate that to a patient and see if you can automatically, within a very rapid period, try to sort of both predict and do experiments to sort of devise what kind of combination is good for this individual person. I'm looking forward to this project a lot. This is something to come. So we have collaborating with many people, thanks to these, and thanks to all our funders. And thank you very much for letting me talk here. And look forward to any questions you might have.